This is Stephen Lewis. I'll be your guide on a brief tour of the animal cell model. The animal cell is divided into two regions. The central core is called the nucleus. The region outside the nucleus is called the cytoplasm. The cell is a membrane-bound and membrane-partitioned structure. Not only does a membrane separate the cell from its environment, but inside the cell are many membranes that create microenvironments. These microenvironments are called organelles and allow for the maintenance of conditions to support different activities inside the cell. Think of the cell as a school building, and these intracellular microenvironments as different classrooms. The membranes that form these microenvironments are like classroom walls, allowing Spanish to be taught in one classroom and cell biology to be taught in an adjacent classroom. Inside the cell, one membrane-bound classroom may produce proteins, another may house our genetic material, another may produce ATP, yet another may contain digestive enzymes to destroy invaders to the cell. The membrane that separates the inside from the outside of the cell is called the plasma membrane. Don't refer to the plasma membrane as a cell wall. In biology, a cell wall is a rigid structure that is present in plant cells outside the plasma membrane. Animal cells do not have cell walls. Instead, our cells have flexible membranes, whose major function is to regulate what can cross into and out of the cell. We call this feature of the plasma membrane selective permeability. The composition of the fluid inside the cell is very different from the fluid outside the cell. This difference in composition is necessary for cell survival and is maintained by the selective permeability of the plasma membrane. If you rupture the plasma membrane, the cell will die. The cytoplasm contains specialized regions called cytoplasmic organelles. These organelles perform special functions inside the cell. The fluid portion of the cytoplasm outside the organelles is called the cytosol. One type of organelle inside the cytoplasm is called mitochondria, shown on the cell model in orange. The mitochondria are sometimes called the powerhouse of the cell because they are the site where most of the energy from dietary fuel molecules is converted to ATP, the type of energy used by the cell to do work. Just as your car requires energy in the form of gasoline, not oil or sugar, the cell requires energy in the form of ATP instead of carbohydrates, fats, or proteins. The mitochondria contain the enzymes needed to transfer energy from dietary fats, carbohydrates, and proteins to ATP. This is why ATP is called the universal energy currency of the cell. Muscle cells may contain hundreds of mitochondria. Note that a mitochondrion is composed of two membranes, a smooth outer membrane and a folded inner membrane. The folded inner membrane creates an enormous surface area to act as support for the enzymes and transport proteins that transfer energy from dietary fuel molecules to make ATP. The Golgi body is a stack of membranes inside the cell where substances produced by the cell can be modified and packaged in little bags called vesicles before they're released to perform their cellular functions. Here is an artist's view of a Golgi body. At the top you can see vesicles arriving at the Golgi body, carrying compounds to be modified. And at the bottom, you can see secretory vesicles being released from the Golgi body after their contents have been modified. Some of these vesicles will end up in the plasma membrane, and their contents secreted to the outside of the cell. One of the vesicles produced by the Golgi body, but retained within the cell, are called lysosomes. The lysosomes are shown in yellow on our model. The word lysosome means bodies that destroy. Inside the membrane of the lysosome are digestive enzymes capable of destroying worn-out cellular parts and bacterial invaders to the cell. The inside of the lysosome is more acidic than the cytosol. This acid environment activates the lysosomal enzymes and helps prevent the enzymes from damaging the cell if they escape to the cytosol. Lysosomes are known as the garbage disposal of the cell. The peroxisome, shown in purple, also contains numerous digestive enzymes. The peroxisome gets its name from its production of hydrogen peroxide, H2O2, which it uses to break down compounds like formaldehyde and ethanol, which you can get from your beer or margarita. The peroxisome also contains enzymes that prepare long-chain fatty acids for use by mitochondria to make ATP. Peroxisomes are a product of the endoplasmic reticulum, which is a vast network of channels throughout the cytoplasm of the cell. There are two major types of endoplasmic reticulum in our cells. The rough, or granular endoplasmic reticulum contains ribosomes in its membrane and produce proteins that usually end up in the plasma membrane or secreted to the outside of the cell. The smooth endoplasmic reticulum does not contain ribosomes and so does not make proteins. But the smooth ER does contain enzymes for the manufacture of many products, especially fatty substances. The ribosomes on our model are shown as white dots. Some ribosomes are attached to the endoplasmic reticulum, whereas others are free in the cytosol. Ribosomes are the sites where proteins are made inside the cell. Our bodies are about 15% by weight protein. Proteins in our food are destroyed in our GI tracts, so the proteins in our bodies must be made in our cells. The centrosome, or central body, is the heart of the skeleton of the cell, the cytoskeleton. The cytoskeleton helps maintain the shape of the cell and can enable some cells to move, such as during cell division. Within the centrosome are two cylindrical bodies called centrioles that are composed of microtubules. The other major region of the animal cell is the nucleus. The nucleus is separated from the cytoplasm by two membranes. Here is a close-up of the nucleus, and you can see clearly its two membranes. The outer membrane is shown in blue and is part of the endoplasmic reticulum. The inner membrane is shown in yellow. The two nuclear membranes are separated by a space, and so the double membrane is often called the nuclear envelope. An envelope has two membranes with a space in between where you put your letter or your check. Pores in the nuclear envelope allow for the exchange of substances with the cytoplasm. Inside the nucleus is an organelle called the nucleolus, shown here in orange. The nucleolus of the nucleus is the site where ribosome subunits are assembled before they're shipped to the cytoplasm where they will manufacture proteins. Also in the nucleus is our genetic material, or chromatin. The chromatin is made of DNA and protein. When the cell is doing its chores, the chromatin is dispersed so that it can be read to make cellular products. When the cell is preparing to divide, the chromatin is replicated. When the cell is ready to divide, the chromatin is condensed into chromosomes, which can be stained and seen through the light microscope. The condensing of the chromatin into compact chromosomes makes it easier to transport the genetic material from the parent cell to the offspring cells. Prior to cell division, the centrosomes are also replicated and migrate to opposite poles of the cell. The two centrosomes produce microtubules that attach to the double-stranded chromosomes and transport copies of each to the two new cells. The result is that each new cell inherits a copy of each chromosome. Thanks for taking the tour of the animal cell model.